The search for extraterrestrial life on Mars is ongoing, and the Mars 2020 Perseverance rover just landed in Jezero crater this past February. This rover is equipped with some new technology for life detection, including a deep UV Raman spectrometer that can detect organic molecules in rocks, and it's capable of sample caching for future sample return missions. This rover is looking for ancient signs of life at Jezero crater. The surface of Mars is extremely cold, arid, and exposed to lethal radiation, and that makes it unlikely that anything is currently alive there. But it is possible that extant life exists on Mars in the deep subsurface. Here, conditions are more stable, offering protection from extreme temperatures and radiation. And there's evidence to suggest that there could be liquid water, which is essential to all life as we know it. And redox disequilibria could provide energy to sustain life. While the distribution of available electron donors or acceptors in the Martian subsurface is unknown, possible substrates include gases like hydrogen and methane produced by water-rock reactions, minerals like iron oxides, or other substrates like nitrate. We humans are not capable of eating hydrogen or respiring minerals, but microbes are. To speculate on what microbes may be capable of in the Martian subsurface, we look at what microbes are doing in the terrestrial subsurface. In rocky subsurface settings on Earth, we know that microbes like bacteria and archaea live in deep, fluid-filled fractures. But this water is anoxic, decoupled from solar energy input, and has very little organic carbon. So how do they survive in such a harsh environment? We know from studying these systems that microbes here can get their energy by catalyzing redox reactions, like iron oxide reduction with hydrogen. Microbes can then use this energy to do other stuff, like divide and create more microbes. But building biomass requires carbon. There's not a lot of organic carbon, so autotrophic microbes get their carbon from inorganic sources, like bicarbonate or dissolved CO2. Using observations like these, we can start to picture what life might look like in the Martian subsurface, but there's still a lot of stuff we need to sort out on Earth to get a better handle on Mars habitability. The terrestrial subsurface can be broadly divided into two settings, the marine and continental. Our research group is focused on the continental subsurface because there's so much we're still trying to understand about this system. The deep continental biosphere is extremely diverse, hosting life across all three domains of life, and it's one of the largest reservoirs of bacteria and archaea on the planet. Over the past few decades, there's been an enormous effort in the science community to understand what makes this system habitable. For example, how are microbes getting their carbon and energy? How much life is there in this system? And what kinds of microbes live here? This work is incredibly challenging because it entails sampling microbes at kilometer depths into the crust. Once you have access to the subsurface, the next step is actually taking samples of the microbes. There's two types of microbes in this system, the microbes that float or swim around through the fluids called planktonic microbes, and the microbes that attach to rock surfaces called biofilms. Most of what we know about the microbes here comes from sampling the planktonic microbes because it's just logistically easier to sample the fluids. Sampling the biofilms on rocks is challenging because if you remove rock from the system, you're going to alter it long term, and so you can't easily go back year after year and keep collecting rock. That means we have to come up with special strategies to study the biofilms without altering the system, and it's extremely important to characterize the biofilm communities because they probably make up the majority of life here. It's currently estimated that biofilms make up as much as 80% of the bacteria and archaea in this system, and if we're trying to understand subsurface habitability on Mars, we need to understand how the majority of life in this terrestrial subsurface system operates. So in 2018, we got funding from NASA to do exactly that, characterize biofilm communities in the continental subsurface. Our Mars Rocky Subsurface Analog Site is a former gold mine in the Black Hills region of South Dakota. Here you're looking at the open cut at the surface of the mine. Now let's take a look below the surface. Using this elevator, we can access levels in this mine as deep as 4,850 feet or 1.5 kilometers below surface. This mine is now the Sanford Underground Research Facility dedicated to subsurface research. The way we access the microbes here is through special borehole installations like this. These boreholes tap fracture fluids in metamorphic rock rich in iron and sulfur-bearing minerals. This is a cross-sectional view of the mine where we've established the Deep Mine Microbial Observatory, or DEMO for short. The gray lines here are the various shafts and tunnels throughout the mine, and these blue dots represent the locations of 6 boreholes throughout the mine where we can access fracture fluids. This is a profile view of a borehole in the rock wall with fracture fluids draining out. We plug these boreholes with installations like this borehole packer, which is basically a plastic pipe with outflow ports on the side. We attach bioreactors to the outflow ports on the borehole installations, and we leave these in the field for a few months to allow microbes in the fracture fluids to colonize the material in the bioreactors. Using DEMO as our Mars analog and our bioreactors to access the biofilms, we can explore our central research question: what role does host rock mineralogy play in subsurface habitability? Specifically, we were interested in exploring how biofilm diversity and biomass compares to the planktonic community. Is mineral selectivity driving biofilm biomass distribution? Are microbe-mineral interactions active or passive? And how important is iron cycling in this system? First, we tackled this question. We wanted to find out whether the biofilms were different from the planktonic microbes, and if so, whether minerals can explain these differences. To explore this question, we picked 3 sites across a range of depths and fluid chemistries at sites DEMO 1, 3, and 6 on the 800, 2,000, and 4,850 foot levels. First, let's take a look at the average fracture fluid chemistry at each site from 3 years of monitoring data with concentration on the x-axis. You can see there's lots of available dissolved electron donors and acceptors in the fluids, which include hydrogen, methane, ferrous iron, ammonia, sulfide, and sulfate, all of which can support a variety of microbial metabolisms. And this fracture fluid chemistry is a result of fluid interaction with the host rock at DEMO. These rocks are rich in iron and sulfur-bearing minerals, which could also be used as electron donors or acceptors. But using minerals as energy sources requires special metabolic strategies, such as extracellular electron transport, where rather than bringing a dissolved substrate into the membrane for redox reactions, electrons can be transported outside of the cell to the mineral surface. And this process requires cells to be in close proximity to the mineral surface. So in this case, the minerals will be a more accessible energy source to the biofilm communities. The amount of available energy for a microbial metabolism is going to affect how much these microbes can divide and increase biomass. And not all microbes can eat the same stuff. So the microbes in the biofilms might be taxonomically different from the microbes in the fluids. So we carried out some field experiments to explore whether minerals drive differences in biomass and biodiversity between the biofilm and planktonic communities. At each of the 3 sites we chose for this study, we set up a series of bioreactors filled with either inert material like sand or filled with crushed minerals representative of the host rock lithology, which include iron, sulfur, and manganese-bearing minerals. We included glass slides in control experiments and polished mineral coupons in experiments with minerals. To compare biomass between the biofilms and planktonic microbes, we estimated cell densities on the mineral coupons and glass slides using scanning electron microscopy, and from the fluids using fluorescence microscopy. And to compare community compositions between biofilm and planktonic communities, we sequenced DNA from each targeting 16S rRNA taxonomic marker genes. We can use what we know about the fracture fluid chemistry and host rock lithology to think about how energy could drive differences in diversity and biomass. Using thermodynamic modeling, we can predict which minerals might be good energy sources at DEMO. This figure shows Gibbs free energy on the x-axis with exergonic reactions to the right of the dotted line. And the various colors denote the different minerals we modeled with the available dissolved electron donors and acceptors. And you can see that all of these minerals can be energy yielding in the right reaction, but some seem to be generally better energy sources than others. If we focus just on the most exergonic reactions modeled, we see that the highest energy yield is from nitrate reduction with pyrite, but the majority of these reactions are with a manganese oxide mineral called pyrolusite. So from this, we can predict that pyrolusite could be a preferable electron acceptor for members of the biofilm communities, and we can test this out using field experiments to see if anything interesting happens when we introduce these minerals to the communities. First, we can look at alpha diversity between the planktonic and biofilm communities. Here I'm showing OTU richness on the y-axis, which is basically the number of different species in the communities, versus phylogenetic diversity on the x-axis, which is basically a measure of how similar those species are. The different colors represent each site, and different shapes represent each community type. This figure is showing that the fluid communities, represented by circles, are generally more diverse than the biofilm communities, which are represented by squares and triangles. And so the mineral and glass surfaces are likely selecting for a subset of the fluid communities. Now looking at beta diversity, this is an NMDS plot showing OTU level Bray-Curtis dissimilarity. Each point on this plot represents a community, and the closer the points are to each other, the more similar those communities are. We see that communities from each site form clusters controlled by the distinct fluid chemistry at each site. We can break this down by community type. These are the fluid communities from each site, biofilm communities on inert control substrates, and biofilm communities on minerals. And we see that there is a good amount of overlap among the community types, with the exception of Demo 1, where fluids and biofilms are pretty different. So which taxa are driving these differences? This is a relative abundance plot showing 0 to 100% abundance on the x-axis, where the different colors denote the different taxa. Here I'm showing the fluid communities from sites Demo 1, 3, and 6. And now comparing them with biofilm communities that colonized inert sand, we see a shift in community structure from fluid to biofilm community at each site. For example, the Demo 1 planktonic community is dominated by Omnitrophica and unassigned taxa, which shifts to being dominated by Rhodocyclaceae in the biofilm on sand. Now we can add the biofilm communities that colonize the various minerals, which are compositionally different from the biofilms on inert sand. In particular, we see an enrichment of these red and black taxa, which represent Desulfobobaceae and Thermodisulfovibrionia, two taxa which are known to reduce sulfate. And pyrolusite in particular seems to promote colonization by these taxa. Maybe the manganese oxide enhances their colonization by scavenging the sulfide produced by sulfate reduction, or maybe these taxa are using the manganese oxide as an energy source, as our models would suggest is highly favorable. We'll come back to this thought a little bit later, but either way, these taxa seem to be responding to this mineral. We can also compare cell morphologies in the biofilms using scanning electron microscopy. Here I'm showing biofilms on glass from each site, next to their respective biofilms on pyrolusite. And we see a shift in cell morphology. For example, the biofilms on glass at Demo 1 are rod-dominated and shift to being coxy-dominated on pyrolusite, indicating a different community is colonizing the mineral over the glass. We also see big differences in cell densities on the minerals relative to the glass, for example, comparing Demo 6 glass to pyrolusite. We actually compiled about 1,300 SEM images of the biofilms to estimate cell densities across the inert control and mineral experiments. Here I'm showing the average cell densities in experiments with inert sand at each site, which had densities between 10 to the 5th and 10 to the 6th cells per square centimeter. And here are the densities on each mineral type, which generally had similar or lower densities than the sand experiments, with the exception of a few experiments, particularly communities on Demo 6 pyrolusite, which are about an order of magnitude higher than communities in experiments with inert sand. And if we assume a fracture diameter of 100 microns, we can convert fracture fluid cell densities from cells per milliliter to cells per square centimeter. And we see that the biofilms are around 3 to 4 orders of magnitude more dense than the fluids. So this is consistent with previous ideas that biofilms likely represent a much greater proportion of biomass than fluid communities in the subsurface. So to recap, generally we found that the fluid communities were more diverse than their respective biofilm communities, suggesting mineral and glass surfaces are likely selecting for a subset of the fluid communities. However, different taxa dominated the biofilms versus the fluid communities, with some taxa representing over 50% of the biofilms, indicating certain minerals promote colonization of these taxa over others. We found much higher cell densities in the biofilms compared to the planktonic communities, and found that specific minerals enhance biomass. And one explanation for the differences in diversity and biomass in the biofilms is that those microbes may be using the minerals as energy sources, as our models suggest. The takeaway from this is that host rock mineralogy is probably an important control on biofilm diversity and biomass, where minerals can host taxonomically distinct communities of significantly greater biomass than the fluids in the continental subsurface. We wanted to take these findings a step further. If microbes are using the minerals for energy, then microbes may be aggregating around energy-rich minerals. And given that mineral distributions in rocks can be very heterogeneous, then biofilm biomass distribution may be heterogeneous too. So we wanted to find out whether mineral selectivity might be regulating biofilm biomass distribution. One reason this is important is that current estimates of biofilm biomass in the continental subsurface are loosely constrained. Biofilm-based bacteria and archaea could make up anywhere from 20 to 80% of the total biomass here. Other studies have estimated continental subsurface biomass using a number of important variables like depth and temperature that are strong controls on microbe distributions. And based on what we've seen so far at demo, we think minerals could also be an important driver of microbe distributions, especially for the biofilms that might depend on those minerals for energy. For example, you can see how heterogeneous this rock surface is. This is one of the rocks we collected from the gold mine at demo, and all these yellow areas here are the energy-rich mineral pyrite, and the dark areas are mostly the energy-poor mineral quartz. If the microbes are getting their energy from the minerals in the rock, then we would expect to see preferential biofilm colonization of the pyrite relative to the surrounding minerals. For example, in the transect outlined in the orange box, we would expect to see a higher density of microbes as we go from left to right onto the pyrite grain. So I ran some more growth experiments in the mine using native rocks to test this idea out. These experiments were installed at sites Demo 1 and 3 on the 800 and 2,000 foot levels. We set up the bioreactors the same way as before, but this time instead of using minerals, we used native rock collected from the mine. We included polished rock coupons in the experiments with rocks and glass slides in the inert control experiments. These were imaged with coupled scanning electron microscopy and X-ray energy dispersive spectroscopy to visualize cell and element distributions on the rock surfaces. And again, we sequenced DNA from biofilms and planktonic communities to compare their taxonomic compositions. We used 4 different rock types in these experiments, which are the 4 major rock units that the demo boreholes intersect. These are called Poor Man Formation, Ellison Formation, Homestake Formation, and Yates Unit. Ellison is a micaceous schist, Poor Man is a pyrite-rich schist, Homestake is iron formation, and Yates Unit is metabasalt. You can see from the bar plot that these are silica and iron-rich, but Poor Man has much higher sulfur content due to the pyrite in the rock. The SEM images here show examples of some of the mineral grain boundaries that we targeted for mapping cell distributions. These are all iron-rich grains. Notice that they're all iron and sulfur except for Yates Unit, which is iron and titanium-rich. These grains we think are pyrite and ilmenite based on geologic descriptions of the rocks from a previous survey. I mapped out elements and biofilm distributions across lots of mineral grain boundaries in the rocks, and here's an example of what that data looks like at about 3000x magnification. In the top panel, you can see the transect across a mineral grain, and below that are maps of iron and sulfur collected with X-ray energy dispersive spectroscopy. Adding these images together, we get the composite image of the mineral grain, and the bright pink dots are the locations of microbes attached to the rock surface. The lines are kernel density contours of the microbes, where the bright yellow contours are the highest density of cells, which you can see occurs on the iron and sulfur-rich region. We mapped coupons from all 4 rock types at each site in duplicate for a total of 16 transects. You can see in these examples the cell densities are higher in the iron-rich regions. The pattern of biofilm distribution looks pretty non-random, and so the next thing we wanted to do is see if we can explain these cell distributions as a function of the underlying element distributions in the rock surface. To do this, we fit spatial models to the data. Here I'm showing an example of one of the transects across an iron and sulfur-rich mineral grain. We fit nested Poisson point process models to the data using null random models that assume homogeneous intensity across the whole area, as well as models with elements present in the rock as covariates. Here you're looking at the predicted intensities across this rock coupon as a function of iron, where the white points are the true positions of the cells and the colors represent the predicted cell intensities where yellow is highest. Now we'll iterate over different combinations of elements in the rock to explain cell distributions until we land on the best fit model. The way we're measuring the best fit is by maximizing this LRT value, which is the likelihood ratio test statistic that gives us a sense of how much better our model with elements fits the data relative to the null model. And we're trying to minimize this p-value, which is a measure of how confident we can be about rejecting our null model in favor of our model with elements. So let's watch how these values change as we iterate over the models. You can see the LRT value is gradually increasing and p-values are gradually decreasing until we land on the best fit model with sulfur and silica. So we fit models to all of our transect data and ended up with these results. We ran simulations of these models and checked how well they fit the observed data, and for the most part they fit the data really well. You can see not all models have the same elements, but if we look at the frequency of the elements invoked across the best models, we see that they all include iron, sulfur, or titanium. And so from this we interpret that these three elements are the best explainers of biofilm distributions. And these elements are associated with the mineral grains we targeted for these transects, where we could see those hotspots of cells. And so it looks like there is a spatial dependence of the biofilms on the iron-rich minerals. Next, we wanted to probe specific taxa that might be involved in mineral selectivity. This is a heat map of correlation showing taxa in the biofilm communities at each site most strongly correlated with iron, sulfur, and titanium in the rocks, where yellow indicates a positive correlation. These Pearson correlation coefficients were calculated from pairwise comparisons of the relative abundances of taxa across each community and elements across the rocks. In Demo 3 biofilms, these two taxa correlate with iron and titanium. There's not a whole lot known about their metabolisms, but maybe these microbes can metabolize the iron and ilmenite. In Demo 1 biofilms, the sulfurous siliceae correlate with iron and sulfur. These microbes are documented sulfur cyclers, and we previously saw them enriched in our experiments with pyrite, so maybe they can metabolize the sulfur in pyrite. And the hydrogenophalaceae correlate with iron and titanium. The hydrogenophalaceae in these communities are dominantly within the genus Thiobacillus, and at Demo 1 they comprise as much as 17% of the biofilm communities on native rock, versus just 2% of biofilms on sand and less than 1% of the fluid community. The Thiobacillus are a genus well known for the ability to oxidize iron or sulfur. They're known to selectively colonize pyrite and can oxidize pyrite with nitrate, which is a highly thermodynamically favorable metabolism at Demo. So maybe that could explain their enrichment in the biofilms. Or maybe they can use the reduced iron and ilmenite as an electron donor in the case of Yates unit. So to recap, we saw the microbes seem to aggregate on the iron-rich regions of the rock surfaces. Our models support the microbes are selectively colonizing the pyrite and ilmenite grains. Our community data showed that the Demo 1 biofilms were enriched in Thiobacillus, a genus known to selectively colonize and oxidize pyrite with nitrate, which is a highly favorable metabolism at Demo. The takeaway from this study is that mineral selectivity is indeed a driver of biofilm biomass distribution. At Demo, we're seeing microbes colonize iron-rich minerals, likely because they're getting their energy from these minerals. And this could be important for constraining biomass estimates at a regional scale, where, for example, regions of the subsurface rich in iron-bearing minerals could support relatively more biofilm biomass. Next, we wanted to investigate other ways minerals might enhance habitability of the subsurface by thinking about active versus passive microbe-mineral interactions. Our models from the first study show that manganese oxide is a favorable electron acceptor at Demo, but these biofilms were enriched in microbes known for the ability to reduce sulfate, which is a metabolism that generates hydrogen sulfide. Hydrogen sulfide will spontaneously react with manganese oxide to form aqueous manganese 2+ and elemental sulfur. If sulfide concentrations get too high, it becomes toxic to sulfate reducers, and it will also thermodynamically inhibit sulfate reduction. So sulfide scavenging by the manganese oxide is one way these microbes could be dealing with sulfide. So our question is, Were those microbes actually respiring the manganese oxide, or could a passive process like sulfide scavenging be enhancing colonization of the manganese oxide? To address this question, we need to do some controlled lab-based experiments. For this study, we collected fracture fluids from Demo 3, located 2,000 feet below surface, and took it back to the lab. We used the fracture fluids to inoculate growth experiments targeting a microbe representative of demo biofilm communities that's capable of growth on manganese oxide. Guided by our thermodynamic models, these experiments were amended with a suite of treatments including favorable electron donors like thiosulfate, elemental sulfur, or simple forms of organic carbon including acetate, propionate, and formate. We also tried to enrich for hydrogenotrophic and heterotrophic sulfate reducers in the presence of manganese oxide. In one of these experiments, we could see something clearly happening with the manganese oxide, the experiments with thiosulfate. We diluted these cultures to extinction to get an isolate, which means we got a single strain of this microbe. And we noticed that as these microbes grew, the manganese oxide disappeared and white precipitates formed, which are presumably elemental sulfur. We tried taking away the manganese oxide to see if they could grow solely on thiosulfate, and they could not. So from this, we could tell that the manganese oxide was enhancing the growth of this microbe, but we needed some more information to get at what might be happening metabolically. So next, we sequenced its genome so we could see what its genes can tell us about its capacity to metabolize manganese or thiosulfate. We found no diagnostic genes for manganese reduction or thiosulfate oxidation. However, we did find that this microbe has the capacity for thiosulfate disproportionation to sulfide and sulfate. This organism also lacks hydrogenases, and so it can't use the hydrogen gas in the culture as an electron donor. Based on this data and the fact that this microbe didn't grow without the manganese oxide, we think that the growth of the isolate is enhanced by the manganese oxide due to passive sulfide scavenging. Recall that not only is sulfide toxic at high concentrations, high sulfide activity will thermodynamically inhibit thiosulfate disproportionation, hence the poor growth without manganese oxide. The model here is showing Gibbs free energy from thiosulfate disproportionation under a range of sulfide activities, and you can see the reaction gets more exergonic with lower sulfide activities. If the manganese oxide is actually spontaneously reacting with sulfide in situ, then microbial reduction of manganese oxide will be kinetically inhibited. Cryptic processes like sulfide scavenging won't be captured by our models, so while metabolisms like manganese oxide reduction may appear thermodynamically favorable, they may not be kinetically favorable. And so these experiments demonstrate the importance of testing thermodynamic models with isolates to get insight into the physiology of microbe-mineral interactions. Now that we have an idea of this organism's functional capacity, what about its taxonomy? Is this microbe actually a good representative of the demo biofilm communities? We carried out a suite of phylogenetic analyses indicating our strain is novel, and it's the third isolated member of the genus Sulphuracella, which we dubbed Sulphuracella species D3-1 after its source site Demo 3. And Sulphuracella is indeed a good representative of demo biofilm communities. Sulphuracella made up as much as 30% of biofilms on hematite at Demo 1, with as much as 4.5 orders of magnitude greater biomass than the respective fluid community. And on native rock at Demo 1, Sulphuracella made up as much as 13% of biofilms on Poorman Formation. This is suggesting potentially significant mineral-enhanced growth of Sulphuracella at Demo, where iron oxides in the host rock may be serving as a sink for hydrogen sulfide. The next thing we wanted to do was compare the functional capacity of the Demo isolate to the two other isolates, both of which come from freshwater lakes in Japan. Unlike the Demo strain, the two isolates from Japan do have genes for iron and manganese reduction, thiosulfate oxidation, and hydrogen oxidation. These differences in metabolic potential might be a reflection of their source environments. Sulphuracella denitrificans SKB26 was isolated from a freshwater lake in Japan called Lake Mizugaki. This lake is rich in nitrate and organic carbon relative to Demo, and these are good substrates to couple to thiosulfate oxidation or metal reduction. In contrast to this freshwater lake, Demo is a ligotrophic, and so nitrate and organic carbon are very limiting. That means competition for substrates like hydrogen, organic carbon, or nitrate may be high, and so a metabolism like thiosulfate disproportionation might be advantageous since it doesn't require those substrates. Although these three members of Sulphuracella were isolated from completely different environments, they do share important metabolic traits that make them resilient under hostile oligotrophic conditions, such as the ability to fix carbon and nitrogen, and they all have genes that make them resistant to toxic substrates like perchlorate and arsenic. And the Sulphuracella don't just occur in Japan or at Demo. You can see from this map we compiled from their distributions that they occur across a wide variety of environments, including marine and both surface and subsurface continental settings. And the detection of Sulphuracella across several other continental subsurface environments, represented by the orange dots, suggests this microbe might be an important sulfur cycler in deep continental systems. So to recap this study, we isolated a new member of the genus Sulphuracella. This microbe is a representative member of deep subsurface biofilm communities. Its growth seems to be enhanced by manganese oxide removing the sulfide generated by thiosulfate disproportionation. This sulfide scavenging is important for keeping sulfide activity low, making sulfide-producing metabolisms thermodynamically favorable. And we see a global distribution of Sulphuracella, including several other deep continental systems besides Demo, indicating Sulphuracella might be an important sulfur cycling member in sulfur-rich deep continental settings. The takeaway from this work is that metal oxides may be important for subsurface habitability, not only as a potential electron acceptor for energy metabolisms, but also as a passive sink for sulfide that promotes sulfur cycling metabolisms. This is especially important to consider given the abundance of metal oxides in deep continental systems and the prevalence of sulfide in deep crustal fluids. Interactions between biofilms and minerals have the potential to further contribute to subsurface habitability by providing aqueous substrates that can fuel the planktonic fraction of the biosphere. So to bring this research full circle, let's talk about one more study investigating the potential for iron cycling among the planktonic microbes in this system. We saw earlier that biofilms on native rock were enriched in taxa capable of nitrate-dependent pyrite oxidation, which is a reaction that releases aqueous ferrous iron. If we imagine this happening at the kilometer scale across Demo, we can picture how bioleaching of iron-rich minerals could be contributing to the extremely high iron concentrations we observe in the fracture fluids, where concentrations locally exceed 8 milligrams per liter. You can see there's so much iron in the fluids, we can see it build up on our filters after we take samples. And given the high competition for energy sources, iron might be a great source of energy for the planktonic microbes too. We can explore iron cycling potential using a technique called metagenomics. What that means is we filter a few liters of fracture fluids, which concentrates the planktonic microbes onto a filter. Then we extract DNA from the filter and sequence all of the genes these microbes have. We can then look at all the metabolic genes in the community and see if any of these microbes are capable of using iron as an energy source. Maggie's former postdoc, Dr. Lily Momper, led a metagenomic study of the planktonic communities at sites Demo 6 and a nearby site called Dussel D back in 2017. These sites are both iron-rich, but no genes for energy metabolisms with iron were detected. There's two reasonable explanations for this. Either there are no iron-cycling microbes at these sites, or the annotation pipeline used at the time wasn't capable of detecting these genes. A new pipeline called FeGenie was just published last year that is highly optimized for detecting iron-specific genes. And so we wanted to test this out and see if we get some hits for iron-cycling genes in a new dataset. We sampled fracture fluids from all 6 Demo sites, and we compared it to Lily Momper's 2017 survey, which includes Demo 6 and site Dussel D. We also sampled a surface community for comparison from Whitewood Creek, which drains into the Demo aquifer system. And we sampled fluids from mine service water used for drilling operations as our anthropogenic in situ control. Before we dive into the metagenome data, let's take a look at the fracture fluid chemistry across the Demo sites to get some environmental context. We can see that sites Demo 1, 2, and 3 are relatively iron-rich, sulfide-poor, and relatively oxidizing, compared to sites Demo 4 and 5, which are iron-poor, sulfide-rich, and relatively reducing. And Demo 6 is iron-rich, sulfide-poor, and very reducing. Based on this, we might expect to see more iron cycling potential at the iron-rich sites, and we might expect to see genes for iron oxidation or reduction to vary with redox potential. Now let's look at a breakdown of the Gene functional gene annotations for the metagenome assemblies from each site. The warm colors here represent genes for iron reduction, and the cool colors are genes for iron oxidation. You can see there are no genes for iron cycling in the surface community from Whitewood Creek, and relatively few genes in our mine surface water anthropogenic control community. In contrast, we see lots of iron cycling potential in the demo communities, where generally there's higher potential at the more iron-rich sites Demo 2, 3, 6, and Dussel D. These genes are dominantly those for iron reduction, however there's relatively more genes for iron oxidation at the shallower, more oxidizing sites Demo 2 and 3. Demo 1 is also iron-rich and relatively oxidizing, and we see dominantly genes for iron oxidation here. Overall, we're seeing lots of potential for iron cycling here, and we definitely see it in the data Lily Mompert collected back in 2017. Next, we wanted to take a look at which taxa have the potential to use iron as an energy source, so we annotated genomes binned from the metagenome data. And generally, we see taxonomic trends consistent with what we would expect for some of these energy metabolisms. For example, we see genes for iron oxidation at the relatively oxidizing sites Demo 1, 2, and 3 in the genomes of Gallinaceae, which is a family of bacteria known for microaerophilic iron oxidation. And we see genes for iron reduction in members of the Desulfobobia and Thermodesulfovibrinia, which are the same taxonomic groups we saw enriched on iron and manganese oxides from our first study. Specifically, the genes we detected in their genomes are involved in extracellular electron transport, supporting the possibility that these microbes can actually catabolically reduce metal oxides. But it's important to notice how diverse these taxa are. Not only do we see the groups we expected to see, but we're seeing lots of other taxa here that aren't the typical iron cyclers you hear about in the literature. For example, across most sites, we see Ignavibacterales, which are known heterotrophs, and some candidate groups like Zixibacteria, whose physiology is unknown. And these taxa have genes for both iron oxidation and reduction, suggesting they might be important iron cyclers at Demo. Now let's talk about what's energetically favorable. Here we're looking at energy density for the planktonic relevant metabolisms with dissolved ferrous iron and ferrohydrate to represent the iron we could see on our filters. The further the points are to the right, the more favorable the metabolism is. Generally, what we can get out of this is that the reactions with nitrate as an electron acceptor or acetate as an electron donor are generally pretty favorable at the iron-rich sites. In contrast to that, we see reactions with methane or sulfide are more favorable at sites Demo 4 and 5, which have higher concentrations of those solutes. And at the shallower sites Demo 1 through 3, we see oxygen reduction is more favorable, and those are the sites that are relatively more oxidizing than the deeper sites. And using another annotation pipeline called metabolic, we do detect functional genes related to oxygen or nitrate reduction, or organic carbon, sulfide, or methane oxidation, and so all of these favorable metabolic pathways are functionally possible. And we can visualize the metagenome data with all the other metabolic processes we detected to show the relative importance of iron cycling at Demo. The previous metagenomic survey from 2017 found sulfur cycling to be a really important process at Demo, but our new data shows that iron cycling is on par with sulfur cycling. So to recap, we found that genes for energy metabolisms with iron align with geochemical conditions, where we see higher abundances of genes for iron cycling at the iron-rich sites, specifically for iron reduction at the more reducing sites, and for iron oxidation at the more oxidizing sites. The specific taxa associated with iron oxidation or reduction genes generally align with our expectations based on their metabolic descriptions in the literature. However, some taxa are potentially important iron cyclers that we didn't expect, so this work sheds light on their potential important ecological role in iron-rich systems. We found that functional potential aligns with our thermodynamic predictions, where energy-dense pathways involving the various electron donors and acceptors we modeled are possible. And overall, the relative importance of iron cycling potential is on par with sulfur cycling potential. The takeaway from this work is that iron-fueled life is probably pervasive in the deep continental biosphere where iron is ubiquitous. And so it's probably important to use optimized pipelines like Gene, where characterizing specific processes like iron cycling in a system is a major goal. This research provides several key insights into continental subsurface habitability. We found that host rock mineralogy is an important control on biofilm diversity and biomass distribution. Minerals can provide an important source of energy, and they can also passively make the system more habitable through processes like sulfide scavenging. And biofilms likely play an important role in subsurface biogeochemical cycling, like bioleaching of pyrite, which releases aqueous ferrous iron that can fuel the planktonic members of the system. Through this work, we learned a lot about rocky subsurface habitability on Earth, and these findings are applicable to the search for life on Mars. We learned that fluid-filled fractures in the terrestrial rocky subsurface host biomass localized in biofilms around energy-rich minerals. The search for extant life in the subsurface of Mars will have to wait for future deep drilling technology. But for now, the search for ancient subsurface life is possible. Perseverance has tentative future plans to visit places where rocky subsurface terrains are exposed around northeast Syrtis just outside of Jezero crater. This terrain is exposed megabreccia that is thought to have mineralized fractures once filled with water. If Perseverance finds one of these fractures, there may be a good chance of finding signs of ancient subsurface life, especially around energy-rich minerals.